Hello everyone, welcome to the Food Tech Networks. This is Astha Sharma and today we are going to discuss about microbiology MCQs, which is a part 3 of this MCQ series. So before starting, if you have not subscribed our channel, please subscribe it for all the latest notifications, please press the bell icon. Let's get started. Question number 1. DASH can be frequently found in humans and animals because of their optimum temperature. And the options are option A, saccharophiles, option B, mesophiles, option C, thermophiles, and option D, saccharotrophs. And the correct answer is option B, mesophiles. Actually, mesophiles have an optimum temperature of around 37 degrees Celsius, which is the average body temperature of humans and animals. So that's why these mesophiles thrive quickly and hence contribute to faster spoiling of food. Some mesophiles are Salmonella and Clostridium perfringens. Question number 2. Some microbes can adapt to low temperatures by increasing the amount of unsaturated fatty acids in the membrane. It's a true or false question. And the right options are option A, true, option B, false. And the answer is option A, true. And the explanation is when the degree of unsaturation increases, it will lead to the dipping of the melting point. Hence, the membranes will remain fluid and the microbe will be able to survive in spite of the low temperature. This can be achieved by reducing the length of the fatty acids too. Question number 3. The reaction rate doubles with every dash rise in temperature. And the options are option A, 1 degree Celsius, option B, 10 degrees Celsius, option C, 10 Kelvin, and option D, 1 Kelvin. And the correct answer is option B, 10 degrees Celsius. So reactions follow the Arrhenius law. This is according to the Arrhenius law, and if we linearize the equation of this Arrhenius law, it will give us a result that a 10-degree rise in temperature doubles the rate of reaction in most chemical and biological processes. Question number 4. Bacteria that are able to survive pasteurization temperature are called option A, halotolerant bacteria, option B, thermophilic bacteria, option C, thermoduric bacteria, and option D, osmophilic bacteria. And the correct answer is option C, thermoduric bacteria. And the explanation is the bacteria that are able to withstand pasteurization temperature of 72 degrees Celsius are called thermoduric bacteria. Halotolerant, halo means salt intolerant, one who can tolerate this salt concentration. So these bacteria can survive high salt concentration, whereas thermophilic bacteria grow and thrive at temperatures above 50 degrees Celsius. Osmophilic bacteria can grow at a relatively high osmotic environment than other bacteria. So thermophiles are subdivided or categorized into like moderate thermophiles, extreme thermophiles. So there are 3 different categories under these. So, and if we one more point over here, halotolerant, I told you, it's related with the salt concentration. There is one more term that is the halophiles. So halophiles, what are halophiles? So these require high salt concentration in medium, whereas halotolerant organism can grow and multiply in the presence of high salt concentration, but these do not require it for their growth. So this is a basic difference between halotolerant and halophiles. Question number 5. At low temperatures, what adaptation do microbes exhibit to maintain membrane fluidity? And the options are option A, osmoregulation, option B, heterovicious, option C, homovicious, and option D, salt regulation. And the right answer is option C, homovicious. Homovicious adaptation refers to the changes made in the lipid membrane so as to maintain the desired cell membrane fluidity. At lower temperatures, this includes increase in the concentration of unsaturated lipids. Whereas at higher temperature, it includes an increase in saturated lipids. In both conditions, low temperature and higher temperature, what the condition is there, increase. What concentration? In low temperature, it's unsaturated lipids, and at higher temperatures, it saturated lipids. And if we talk about the osmoregulation, it is the active regulation of osmotic pressure of an organism body fluids to maintain homeostasis of that organism. And of course, the water content also. That is, it maintains fluid balance and the concentration of electrolytes. Question number 6. What is the optimum temperature range for thermophiles? And the options are option A, 40 to 45 degrees Celsius. Option B, 20 to 30 degrees Celsius. Option C, 60 to 90 degrees Celsius. And option D, 55 to 75 degrees Celsius. And the correct answer is option D, 55 to 75 degrees Celsius. The optimum temperature range for thermophiles is 55 to 75 degrees Celsius. But if we talk about the minimum temperature range, then it is the 40 to 45 degrees Celsius, which is the option A. And if you talk about maximum temperature range, then it is 60 to 90 degrees Celsius. That is option C. So this is because I have already told you, because thermophiles are divided into this medium thermophiles or extreme thermophile. Low is also there. So 3 categories. That's why it like optimum temperature range and minimum and maximum temperature range over there. So question 7. The heat resistance of a microbe is directly proportional to the incubation temperature. So it's again a true or false question. Option A true, option B false, and the correct answer is option A true. Genetic selection and the mechanisms to keep the microbe alive activate the heat resistant genes to function more when the temperature is increased. For example, S. Sandberg was 3 times more heat resistant at 44 degrees Celsius than at 35 degrees Celsius. So thermophilic bacteria, if you talk about those, then they can also thrive in extreme heat because their proteins have an abundance of disulfides. Covalent bonds between these sulfur bonds are there. And one example of bacteria, thermophile actually, which can thrive at it can survive at temperature up to 122 degrees Celsius is Methanopyrus candleri. So it's one of the example, and it can survive at 122 degrees Celsius. Question number 8. Psycherotrophs are also called psycherophiles, psycherophiles. Options are obligate, option B ordinary, option C facultative, and option D complex. And the correct answer is option C facultative. So what happens over here is psycherotrophs or facultative psycherophiles have a minimum temperature range same as obligate psycherophiles, that is -5 to 5 degrees Celsius. However, their maximum range is higher than that of obligate psycherophiles. The maximum range for psycherotrophs is 30 to 35 degrees Celsius, whereas for psycherophiles, it is 15 to 20 degrees Celsius. So that's why they are also known as a facultative psycherophiles or false psycherophiles. So next question number 9. It's a graph-based question actually. It's a growth rate versus temperature graph. And the question is labeled TA, TB, and TC. So the options are TA, you can see over here. Where's the cursor? This is a TA, this is a TB, and this is a TC. So the options are option A, TA, optimum temperature, TB, minimum temperature, TC, maximum temperature. Option B, TA cardinal temperature, TB maximum temperature, TC minimum temperature. Option C, TA maximum temperature, TB minimum temperature, TC cardinal temperature. Option D, TA minimum temperature, TB optimum temperature, TC maximum temperature. And the right answer to this question is option D, that is TA, which is over here, is the minimum temperature, TB is the optimum temperature, and TC is the maximum temperature. So this is, as I told, it's a graph of growth rate versus temperature. One can see that as the temperature begins moving towards the optimum temperature, that is from here, um, from here to here, when the temperature is moving towards this side, that is the temperature is getting higher. So the growth rate is at peak at the optimum temperature over here. Later it starts decreasing as the temperature deviates from the optimum temperature. So the slope becomes steeper as the temperature increases after the optimum temperature. This signifies a rapid lowering of the growth rate. As the temperature decreases from the optimum temperature, the growth rate decreases slowly due to the shutting down of the enzymatic systems in the microbe. Hence, this graph is asymmetric. Question number 10. Bacteria possess the property of dash for communication. Option A, quorum sensing. Option B, contact inhibition. Option C, mobility. And option D, apoptosis. And the right answer is option A, quorum sensing. Quorum sensing, it's a regulation of gene expression in response to fluctuations in cell density. This is possible with the help of autoinducers, which increase as a function of cell density. Contact inhibition, if you talk about, then it's a property of normal cells actually, and it helps cells to differentiate properly, or not allowing cells to get stacked up, or proper multiplication is also there, and this controls everything. And if this contact inhibition, because of any gene mutation or any other underlying cause disrupt, then the cells will not multiply in their in the way they are meant to, and this can lead to um, cancers or tumors, we can say. So mobility, we all know it's the ability to move of any organism to move, either by flagella or whatever means are there. So option D, apoptosis, we all know it's a programmed cell death. If it's a natural process which is going on in our body, if the cell is damaged and it cannot be repaired, or it's aged, or it's dying, then apoptosis or programmed cell death is over there, and automatically that cell gets destroyed. So question number 11, specific regions of bacterial genetic material containing virulence genes are called. Option A, pathogenicity oceans. Option B, pathogenicity islands. Option C, ecological oceans. Option B, ecological islands. And the correct answer is option B, pathogenicity islands. So pathogenicity islands are taken up by the bacteria through horizontal gene transfer. They are found only in pathogenic organisms and are usually present in both gram-positive as well as gram-negative organisms. 2 points over here you should remember how these are transferred. Horizontal gene transfer is there, and they are present in both gram-positive as well as gram-negative organisms. Question number 12, again a true and false question. Pathogenesis is a biological mechanism of a disease. Option A true, option B false, and the correct answer is A. Obviously, pathogenesis can explain the origin of the disease as well as the mechanism of the contracting the disease. It can describe the morphological characteristics of the disease as well as its intensity. Sorry. So question number 13. The study of causation is called option A, rheumatology, option B, histology, option C, otology, and option D, ethology. The correct answer is option D, ethology. Ethology is the study of the origination of something. In medicine, various factors are considered to find the cause of the disease. The disease is said to be idiopathic when no etiology is confirmed. What is rheumatology? Rheumatology, it is the study of food. Histology, it is related with the tissues. It's a study of tissues. And otology, it's a study of anatomy and physiology of ear. Ear are sense organ. Okay, question number 14. Dash curve is used to measure the incubation period. And the options are option A, etiologic, option B, progressive, option C, epidemic, and option D, discrete. And the correct answer to this question is option C, epidemic. An epidemic or an epicurve can be used to predict the incubation time of an organism. The time trend, irregularities, mode of spread, etc. can be predicted from this curve. Question number 15. Salmonella cells are option A, gram positive and sporulating, option B, gram negative and sporulating, option C, gram positive and non-sporulating, option D, gram negative and non-sporulating. And the right answer is option D, gram negative and non-sporulating. These are rod shaped and are gram negative and non-sporulating. And these are facultative anaerobes and are motile. Question number 16. The bacterial cells cause infection in the option A, stomach, option B, small intestine, option C, large intestine, option D, pancreas. And the right answer to this question is option B, small intestine. The salmonella cells attack the small intestine and produce toxins in the mucosa. This leads to the inflammation of the intestine and further gastrointestinal problems. Question number 17. Toxoplasmosis is caused by a option A, bacterium, option B, virus, option C, protozoa, option D, fungus. Correct answer is option C, protozoa. Toxoplasmosis is an infectious disease caused by protozoa Toxoplasma gondii. It can be classified as a parasitic disease too. The disease can spread through insect bites, blood products, or contaminated food and water. Contaminated water is there, and if we talk about food, then it is mostly related with the contaminated pork and seafood. And if I talk about blood products, so it means the blood transfusion, but it is very rare to get this uh, toxoplasmosis via any blood transfusion or blood product. Though it's rare, but can happen. So the point is because before any blood transfusion, the blood is generally screened for different diseases, bacterial infections like HIV, HCV, and other bacterial infection if it is over there. So it's very rare to get toxoplasmosis via blood products or blood transfusion. But contaminated food and water is at the top list, and in US it is very common over there to get and this toxoplasmosis we have contaminated food and water. It is a foodborne illness. uh, this is the first cause over there on the top list. Question number 18. Dash are responsible for acute infection. Option A, tracheozoites, option B, bradiozoites, option C, cytozoites, and option D, vacuozoites. So this is all these stages are related with Toxoplasma gondii or of Toxoplasma gondii only. So the right answer is option A, tracheozoites. Tracheozoites are motile forms of the protozoa. They have the ability to produce tissue cysts during the infection. They enter cells and multiply continually to fill the host cells with the next generation. And the radiozoids, these are quiescent parasite. Quiescent means these are in a state of inactivity or dormant. So these are found in intracellular tissue cysts, found within muscle cells, and within cells of central nervous system, predominantly in neurons. So radiozoids are forms of slow multiplication, and these are these causes chronic infection. The radiozoids found within the tissue cyst are known as the cytoloids. That is option C. And tetrazoids are asexual of this, Toxoplasma gondii. So, question number 19. Cats are more susceptible to toxoplasmosis than frogs. Option A true, option B false, and the option correct answer is option A true. Frogs are cold-blooded animals, whereas cats are warm-blooded animals. Toxoplasmosis affects warm-blooded animals. Also, cats are the host of this parasite. The parasite's eggs can only reproduce inside the feline's body, hence the result. Question number 20. A four-fold increase in dash indicates acute infection. Option A, IgM. Option B, IgG. Option C, leukocytes. Option D, platelets. So these are the antibodies found in our body. And the correct answer to this question is option B, IgG. Immunoglobulin Ig binds and neutralizes toxins. It can activate the pathway used to eliminate pathogens, and increasing this indicates infection. IgG is the most common antibody and it is found in the bloodstream. IgG is the one more thing over here is the only antibody. IgG, it's the only antibody which can cross the placenta because of its size. It can cross no other antibody than IgG can cross the placenta because others are larger in size, and this provides passive immunity to the fetus. And if I talk about the IgM, when it is the first antibody which gets secreted by the adaptive immune system whenever our immune system encounters any antigen. So, thank you. That's all from my side. Do like and share the video. Thank you.